Hello, everyone. My name is Max Chin, and I'm an undergraduate researcher from the Rosenthal and Blackmon labs in the biology department here at Texas A&M. Today, I'm going to be talking to you guys about a project that I've been working on under the undergraduate research scholars program, looking at morphological trends and experimental and simulated populations of hybrid Xiphophorus swordtail fish. So, for today's presentation, I'll be focusing on the simulation portion of this project, just because it's an extremely broad project, which many people in my lab have contributed to, and I would like to kind of discuss some of the more novel aspects which I have personally designed and implemented. So I want to start by giving a brief background on hybrid zones and why they are so relevant to modern evolutionary biology. So to just define some basic terms, what do we mean when we talk about biological hybrids? Well, in the context of this project, we can think about hybrids as being the outcome of genetic exchange between two distinct species. When these events occur naturally, they often do so along what we call hybrid zones, which are essentially geographical gradients between two parental species, with hybrids predominating in transitionary areas. Hybrid zones are interesting to us as biologists as they allow us to observe the process of population level differentiation as it occurs. And they also provide insight into the factors that may guide such differentiation. Two of the most commonly cited such factors are natural and sexual selection. The latter is especially interesting given the incorporation of two distinct traits which are expressed in one sex or the other, namely preference in females and sexually selected traits in males. A fundamental concept describing the mechanism of directional preference and trait coevolution at the genetic level is that of Fisherian runaway selection. This model describes how non-random inheritance of genes associated with these two traits can lead to the propagation of exaggerated sexually dimorphic traits present only in males, with a commonly used example of this being the tail feathers of male peacocks, as you can see here. Keeping all this in mind, one of the primary goals of this project is to explore the effects of natural and sexual selection on newly formed hybrid populations, both in isolation and in conjunction with one another. We accomplish this by computationally simulating these hybrid populations under a variety of evolutionary scenarios, with life history traits being drawn from our model system to inform these simulations. Every research question needs an appropriate model system, and in this case, the Xiphophorus hybrid complex we study is ideal for several reasons. Firstly, the two parental species, Xiphophorus malinche and Xiphophorus birchmanni, are differentiated morphologically, physiologically, and behaviorally. In terms of this project, the most relevant of these differences are the presence of sexually selected caudal fin extension in malinche males, species-specific expression of traits under natural selection, specifically thermotolerance, and finally, species-specific female preference for male sexually selected traits. Additionally, recent studies have provided us with some baseline information on the genetic architecture of these naturally and sexually selected traits, which allows us to design more accurate genomes, which can be further incorporated into our simulations. Finally, long-term research in our lab has provided us with ample empirical data from experimental mesocosm hybrid populations encountering three basic types of ecological conditions, allowing for comparisons to be made with our simulated data sets. Our simulations were designed and run using a program called ADMixM, which is a forward time population simulator developed by our lab, which allows for easy incorporation of genomic architecture and selection functions. To mirror the structure of our empirical data set, two parental populations and three hybrid populations were simulated, with the theory being that each hybrid population encounters a different ecological environment. Each hybrid population was seeded with 10 males from parent population 1 and 10 females from parental population 2, with no further migration occurring between these hybrid populations and populations being simulated for approximately 10 generations. Our simulations incorporated 2 to 3 additive polygenic traits, with a sexually selected sword and naturally selected thermotolerance trait always being simulated, and a preference trait being added for simulations which included sexual selection. Where possible, these traits were incorporated into our genome with reference to published genes associated with the traits. Trait values for the traits could range between 0 and 1, and were fixed at 0 in parental population 1 and at 1 in parental population 2. To explore the effects of natural and sexual selection on our trait values, our simulations were run with a variety of different selection functions, which can be broadly categorized into two main groups. Our natural selection functions determine the fitness of an individual based on its naturally selected trait value, drawing a Gaussian function from this value, with the function being shifted along the x-axis depending on the hybrid population in which it is being implemented. The strength of selection, or the fitness deficit between the most optimally and most minimally fit individuals, was also altered using a selection coefficient, which was incremented between 0.5 and 1. Here you can see how in hybrid population 1, individuals with a trait value of 0 are most optimally fit, while in hybrid population 3, individuals with a trait value of 1 are most optimally fit. On the other hand, our sexual selection functions were kept consistent throughout all of the populations. They instead change in shape based on the preference trait value of the chooser, or female, which is evaluating a mate. In this way, the fitness of a male is, with respect to mate choice, is dependent on both his sexually selected trait value and also the preference value of the female which he intends to mate with. Here you can see an example of a selection function which was designed to represent directional selection on our SWORD trait in our model system. Here you can see how females with a preference value of 1 experience this apathy-like selection for the SWORD with no preference at all, while females with a preference value of 0 experience strong antipathy for the SWORD, preferring males which do not have this trait. Overall, 52 different parameter combinations were simulated, with natural selection functions being implemented in both a consistent and seasonal manner, and 3 different types of sexual selection functions being implemented. Each parameter combination was also run for 100 times to account for stochasticity built into the population simulator. I'm now going to spend some time going over some of the preliminary results that we have that have arisen out of our analysis for our simulated data. And firstly, we tested our hypothesis that our drift-only model, without any selection function being implemented, would show no significant differentiation between the 3 hybrid populations. As you can see in this box plot on the left, this hypothesis is supported by our collected data, which shows that no major trends are occurring over time for either trait simulated. Shifting from a drift-only scenario to one including just natural selection, we expect to see significant differentiation in our naturally selected thermal tolerance trait across dependent variables of both time or generation and site or population. This is the exact trend that we see in our simulated data, with the lowland site drifting towards 0 over time, the highland site drifting towards 1 over time, and the intermediate site or intermediate population remaining fairly consistent over time. You'll notice that much of this differentiation is present even in generation 1, and this is due to how the simulation program essentially implements the selection function prior to scoring phenotype values. Interestingly, if we go over, if you take a look at our box plots depicting SWORD trends in this same natural selection only scenario, we can see that these trends in thermal tolerance are also observed here on a smaller scale, despite there being no actual direct selection on the SWORD trait. Now, this is most likely due to physical linkage with genes contributing to thermal tolerance, which are being selected for, which is expected given the highly polygenic nature of both the traits in the simulation. Now, when we move over to SWORD index trends under directional sexual selection, we see that this bears resemblance to the previously discussed random trends under drift. This can be traced back to how the same sexual selection function was implemented across all 3 populations, and as such, we don't really expect any differentiation to occur. Our statistical analyses identify time as being a significant effector on trait value, and this is in part supported by our plots, which show that there's a slight bias towards a negative decreasing trend, which is in contrast to the previously discussed drift plots, which are approximately equal between increasing and decreasing trends. However, the trends we notice here in our sexual selection scenario are noticeably lower than what we would expect, which probably suggests that not enough time has passed for runaway mechanisms to significantly influence these trends. However, once we combine this directional sexual selection with natural selection, we see that differentiation in sword length is more readily apparent. Comparing these trends against those under just natural selection, we can see that the addition of directional sexual selection has little to no effect, and that trends are primarily driven by physical linkage with genes under natural selection, so our thermotolerance genes. Overall, our preliminary results suggest that all aspects of our simulation design are functioning as planned, with the expected trends observed under drift and selection scenarios. Based on the high level of differentiation associated with natural selection and an apparent lack of significant trends associated with sexual selection, we identified the former as being a key driving force in the differentiation across our early generation hybrid populations. Our next goal is to compare these simulated data sets against our empirical data sets, which has actually already been done, but unfortunately I don't have enough time to go over those results in this presentation. Additionally, we also plan to run those baseline parameter combinations for extended generation time, as well as with reduced population sizes as well. Finally, I would like to take some time to send out some acknowledgements, firstly to both my labs, the Rosenthal and Blackmon lab, for being so supportive and just providing the best possible learning environment for me. The Undergraduate Research Scholars Program for giving me this amazing opportunity to do to engage in something I'm really passionate about. Texas A&M University, of course, our CCHAS field station in Mexico, where most of the data, empirical data that I used for this study was collected. And finally, the National Science Foundation for funding this project through their long-term research and environmental biology grants. Thank you guys so much for listening. If you have any questions at all, please feel free to reach out. I'm more than happy to talk more about this stuff. And finally, if you're ever interested, I have some references that you guys can look up a little bit more as well.